Okay, thank you. Welcome everyone. I'm privileged to be able to tell you something about choosing a relevant model to mimic human diseases. I'm not going to give you answers. I hope to raise a discussion on this really important matter. Briefly about the outline of my presentation, I couldn't come up with a way to talk about this issue without touching the ethics. So I'm gonna go a bit into that. Then of course we're gonna talk a lot about translational research, a bit what it means and where it hopefully will bring us. Then of course some examples or details on choosing the model and some species characteristics on mice and pigs. So I come from Kuopio and my background is in cardiovascular gene therapy, and most of my examples will be from that field, naturally. So are there many in vivo researchers in the audience? Do you do in vivo work with animals? Okay, a few at least. So to touch the ethics a bit, everyone doing animal work, in my opinion, should touch this question at least once. So to stop to think the moral value of an animal. There are no right or wrong answers because it's ethics. We're going to talk about two completely different views and then the middle ground. So first of all, there are people who are strictly against animal experimentation, any kind. They think that that all animals are equal to humans, and thus we do not have any reason to have other rights than animals have. Then there are people who are for animal experimentation, and these people view that the moral status comes from ability to pursue specific life goals, and thus people or human and animals do not belong to the same moral community, and thus they think that we do not have any obligations towards animals, and that always the good arising from the animal experimentation outweighs the bad that it brings. And then the middle ground, people who think that or feel uncomfortable with animal experimentation, but still think that in many cases the good arising from the animal experiments outweighs the harm to the animals. These people think that, or they do not like doing animal experimentation, they want to be as kind as possible, they want to do things as good as possible, and they think that causing cruelty to animals will evidently flaw the moral of human beings also. And then there is the common sense view. So if you ask the question, when the needs of animals and humans come to conflict, which take precedence? So which would you harm if you would need to choose? Rafael Frias, a veterinarian from Turku Laboratory Animal Center, has put it nicely, I think, saying that using animals in research is a privilege given to the scientific community from the society. And when you think about this, it gives a huge amount of expectations to us as scientists. So it brings responsibility on how we do things and what we do. It brings expectations that the experimentation is done in a human way, and of course expectations to get results once we use the animals. You all know we work under the directive on the protection of animals used for scientific purposes. It's a rather new thing. Implementation was done in 2014, and you need to know that the ultimate goal of this law is the full replacement of animals in experimentation. We, as researchers, know that it's maybe not really realistic, but that's the ultimate goal that is written there. And the use of alternatives whenever possible is a legal obligation. It's not voluntary. And of course, the legislation is based on 3 Rs. Then about the translational research and what it means. It's a hot topic, of course. Everyone has heard about it. In my opinion, it means science from bench to bedside and also back. So we actually have this feedback loop, or we should have it. It comes to reality nicely, I think, in Jivas, in using Jivas data from patients and using that to actually check things in animal models. But also we should have that loop after probably failed clinical trials, and we will talk about that later on more. It has been said that all models that try to model human diseases are bad, and some are useful. Of course, this goes with all modeling. So all in all, there is no such thing as perfectly mimicking model, and this is a fact that we just need to cope with. So we can ask why have majority of gene therapy clinical trials, at least in cardiovascular area, failed to reproduce the positive results from preclinical phase. And ultimately, are we modeling even the right things, or are we using the right models? So then some basics on choosing the model. So of course we should start with thinking, what is our research question? So do we actually need a complex model? So if your question is or concerns, for example, fine details on protein trafficking, in complex in vivo model might bring you only more questions, and in vitro modeling might be better. Of course, you would then check it in vivo, but to start with. And then you should think whether you are asking what is the drug, for example, doing to the animal, or what is the efficacy of the drug. These are two completely different things, and you should choose your model according to the question. And by the ethics, of course, you should use the simplest model available. So start from the bottom in the development, if you think about animal species. If you are studying a new gene, just basics of the gene, most probably a genetically modified animal model already exists, and of course it's usually a mouse. And these are commercially available, so you should just seek for them. And when you're planning to model a human disease in animals, you should do a thorough literature search to see what kind of models are used. For example, on hypertrophy and heart failure, there are several models. You can use aortic banding, angiotensin II, valvular defect models, and all of these are used in a bit different settings usually. So you just need to see what has been done and what is the kind of the golden standard model, and there might not be any good reason to change that model. So do not reinvent the wheel if it's not necessary. So then, if you go to the mouse, mouse is usually the first mammalian in vivo model for many studies. This is true because there is a 95 to 98% genetic resemblance to humans. And now we could stop to think, what does this actually mean? We just recently wrote a review on atherosclerotic animal models, and one of the reviewers was really angry at me when I used this phrase, saying that it's completely irrelevant. I think what we want to say with that is that, yes, there is a remarkable similarity in the genetics if you think how different we are actually physically from mice. But still, if we round up and think that there is around 30,000 genes in the mouse genome, it means that 600 to 1,500 genes are different. So you should know whether the genes that you are actually studying or the genes that affect the disease you are studying are actually the ones that differ. All in all, the basic anatomical structures are rather similar in mice and men. Of course, there is the ease of genetic manipulation, and this is really one of the key points in using mice. Because, as said, many of the models are commercially available, and it's rather easy to use the newest techniques with the mouse genome. And then we have the inbred strains. There are several, and they are really well characterized. And by this, you can decrease the genetic variation between your individuals, of course. They say it's 99% or even 100% identical within the inbred strains. But all in all, of course, you need to bear in mind the profound differences, which are, for example, metabolic rate and the physical size, of course. And these then, how would these things then affect the disease or the gene that you are studying? Then to some more specific examples, we have used a lot mouse as a model of atherosclerosis, obviously. So the development of the disease is rather different, of course. The mice do not naturally develop plaques, so we need on the bottom we need the genetic manipulation. And once we have that, and we maybe give high-fat diet, we get rather occluded vessels within weeks. While in humans, it's of course a lifelong development usually. The morphology of the detailed morphology of the plaque differs somewhat, and the place where the plaques are. So mice do not have plaques in coronary arteries, or we cannot study them because obviously the vessels are so small. And still, the current mouse models are more modeling the development of atherosclerosis rather than the complications that are usually the clinical manifestations. Of course, the search for vulnerable plaque model, for example, is really ongoing, but this is the situation at the moment. And then, if you think of heart diseases, of course, mouse heart beats 550 to 750 beats per minute. This is rather different. Here, the metabolic rate has a huge effect, of course. Some parameters of the heart function are scalable with the body mass, but not all, and there are some nice review articles on this. But still, the basic anatomy of the heart is remarkably similar. There are some differences in the ion channel expression, so you might see here there are differences in the action potential, this being the mouse and this being the human, and in the ECG. And then there is the thing that the mice have an enormous ability to recover. So this is a mouse heart 14 days after left under descending arterial ligation, permanent ligation. And this is the left ventricle, this is the right side of the heart. So basically, I would say 60% of the left ventricle wall is a fibrotic scar. This mouse had an ejection fraction of 8%, but it did not have any welfare problem. So nothing was stated. It moved nicely, it was happy camper. And you all know that if a human heart looks like this, there is a severe welfare problem. There is, in European Society of Cardiology meeting just a few weeks ago, there was a nice presentation by Peter Pasterkamp on genetic validation of the mouse data, of all the data that has been obtained. For example, in this specific case, it was on certain atherosclerotic pathways that are associated with altered black phenotype in mice. So they went through actually over 2,000 articles, published articles, and picked up the genes that had popped up there. And then they validated these pathways on GWAS data, available GWAS data on patients with coronary artery disease. And what they found was that, for example, in NF-kappa B pathway, there was 46 genes that were associated in altered black phenotype in mice, and 6 of these came up in the GWAS data. So he just wanted to point out that you should be really careful on selecting your targets. And I think this would be a nice way to actually see, before you start your detailed research, to just see the available data on the databases to see whether it is false positive or whether you might have something to study. Then there are a lot of things to consider when we still use the mouse. So in drug studies, they generally tolerate really high exposures to drugs. They live in stocks. You need to remember this. If you have them single house, they will be miserable. And then to avoid bias, these are just a few things that you should take into account. You should randomize, of course, the individuals in the groups, but also gauge location, sampling order, treatments. You should remember that all the odors, perfume, shampoo, soap, laundry detergents, affect the reproductive cycle. There is a huge difference whether you take your samples during weekdays or weekends. There is different amount of human activity in the animal facility. It makes a huge effect. The cage change cycle. When the cage is changed, it's like an earthquake to the animals. It has been studied that at least 5 hours the activity is increased. Of course, the heart rate is increased. This has a huge effect on the hemodynamics. So you shouldn't take blood samples at least within that day. Then the urinal cycle. You should block the experiment so that you can take all your samples within 2 to 4 hours. And if you actually consider all of these, you realize that you end up with having maybe 2 days in a week that you can actually do your sampling without causing bias. So you should really, really think how to do it. These points were from an issue on International Laboratory Animal Research journal last year. The whole issue was on experimental design and statistics. Well, then of course the translational value of a mouse model is limited. So we will end up on the fact that we should use a large animal model. As said, they have an outstanding recovery abilities and compensatory mechanisms. It is a good model to start with. It is a good model to study the effects of specific genes and maybe even the mechanisms and of the development of the disease, because the genetic alterations are so easy. But then once you go to the therapeutic phase, you should use large animal models also. And that brings us to pig. I'm gonna talk only about pig because this is familiar to us. We use domestic pig as a large animal model. And what makes it a relevant model? Well, again I say the genetics is similar, closely similar. But the anatomy and physiology are even more similar if you compare to mice, of course. We have now the possibility to study actually the coronary vessels rather than the larger vessels. Also, the ethical concerns when using a domestic pig are of course minimal if you compare to non-human primates. That's a fact that comes from using this as a food or growing pigs to food. Many diseases develop in a similar manner in pigs and humans. The size resemblance has huge effects. You can use the same equipment. You can actually mimic the clinical situations really nicely. And then, as a positive thing, there is the genetic variation. So now we are moving away from the inbred strains. We have individual variation, which of course the patients have also. So we are getting closer to the clinical situation. There is beginning to be rather good ways to do genetic manipulation to pigs also with the new systems. And they are actually cheap to buy as experimental animals, but of course they are expensive to maintain. This is to tell you about the translational value of the porcine models. So this is an treadmill model that we use, and we use simply the same treadmill that is used by humans. We can even use the exercise belt to get the ECG signal. And they are rather, no, it should be here, but it's not. But they are really easy animals to teach how to actually go and run simply with the popcorn. And you don't need to use any cruelty, because once they are not coming for the popcorn anymore, then they are too exhausted. So it's a greedy animal. It wants the treat. If it's too tired to come and get it, then we terminate. So it's a nice way to increase the translational potential. Here is the comparison of mouse, pig, and human myocardial scar, in which I find a lot of similarities, and I hope you'll find it too. So there are fibrotic, or there is fibrotic tissue and some inflammatory cell infiltration. So there is potential in all the models to some extent. Then some examples on atherosclerotic lesions. So these are, of course, this is a aortic root from mouse, and then a rapid abdominal aorta and big coronary artery. And you see that there are the same structures to be found. And then something with rabbit and pig, and also there are the same structures to be seen. So to wrap it up, the ultimate question, why have the clinical trials, and I'm safe to say at least in the cardiovascular field or gene therapy of cardiovascular diseases, what are the reasons why we have not translated there? So we could ask, are we modeling the right thing? So is the model that we have used, is it modeling the development of the disease or the manifestations? Are we modeling the what is the drug doing to the animal, or what is the drug efficacy? Are we modeling metastatic cancer with one subcutaneous tumor? So you should really be precise. What is the model you decide to use? What is it valid for? Then are we using the right models? This is the old phrase on studying disease or studying, for example, angiogenesis in young and healthy animals, whilst the patients are really often like non-option patients. So this is something that we really need to consider, and tonight there is a nice poster, number 37, from Galina Wirth about this, and she has been studying angiogenesis in diseased and old animals. to see what is actually the difference. Then there is the fact of prior truck exposure. Usually the patients really have a lot of prior truck exposure, which the animals might not have at all. And of course differences in the truck tolerance. As I said, mice, for example, tolerate a lot higher doses usually. And then the efficacy bars. And this is a difficult one. We set statistical significance bars or some kind of artificial efficacy bars. While these might not be true in the clinical situation, so it's not the same thing as clinical or biological significance. So we might reach a significant decrease in tumor size, but it might not tell the whole story in the clinical situation. Then the amount of things we can control, and this is a huge issue. In the lab we can control a lot of things. We have inbred strains, we have everything, and as said, we should move towards the more variability in all of these things, in the genetics, in all drug exposures and everything, because that is the situation in the clinics. Then meaningful starting points. So to see whether, if you study cancer drug or a tumor suppressing drug, it's not clinically relevant to start your treatment at the same time point when you implant the tumor. Then you're not studying the right thing. So this is as important as choosing the endpoints or the efficacy bars. And then about the delivery of the therapy agent. You should be able to use the same methods, use the same delivery route. You should be able to target the same cells in preclinical phase and what you're planning in the clinical trials. There might be completely, there are huge differences in the transduction efficiency when you compare the animals and the humans in the clinical trials. And there might be unknown mechanisms affecting the transduction of the virus that we don't know yet. And there is a nice work by Anssi Laine in the poster session tonight about this. It's poster number 44, if I remember correctly. So all of this brings us back to the feedback loop. So once the clinical trials, or if they fail, then you should come back, you should see what went wrong, what was wrong in the models if the results were not the same. And take-home message from me is that a model system always remains an approximation, but we are doing the best we can with the tools we have. And then that you can succeed or fail in your research only due to the chosen in vivo models. So you should really do your literature research. You should really stop to think what is a relevant model to use. And by that, I thank you for your attention.
Thank you very much for this very interesting talk. I think it's also very important because, as you outlined in the beginning, it's really an EU directive to avoid animal models at least as much as possible. The talk is open for discussion. Question?
Thanks, Jenni. Very nice presentation. I just wanted to, you were saying about the gait change and everything affecting the mice behavior and possible results on studies. Is this something that you only feel happening with the mice, or is it also a problem with the large animals? And how are we supposed to take all of these into account also with the larger animals?
Well, it mainly depends on the natural behavior of the animal, the natural needs. For pigs, it's a huge thing. If they are housed alone, they are really, really stressed. Rabbits, well, they are more used to being alone, but they are then even more sensitive to the stress. So it all comes back to knowing the species-specific characteristics to know the natural environment where they should be and how you can mimic that in a perfect way. Of course, the hierarchy on the stock then has a huge effect, so you shouldn't change it. That goes with all the species.
Further questions? Perhaps I would have a last question because it's an educational session. Could you comment on the size of the cohort? So if you make a typical experiment with pigs, so are we talking about 5 animals or 50 animals? I know there's big variation for sure, but if you could say, okay, so an average one.
Well, we have a lot of history on doing similar kind of trials with the large animal models, so we know rather well the variation and we know the power that a certain size of a group gives us. So what I would say we normally use is 6. 6 in group.
Okay. Thank you again.